Welcome everyone. Today we would be discussing about structural databases. In the previous lecture, what we have talked about that what is bioinformatics, what are the types of databases, what are their main examples of that, what kind of databases are genomic databases or protein databases. So today we would be focusing on structural databases. That means the main concept of structural database would be discussed, that these databases are basically, as the name suggests, these databases will mainly comprise of the structural information of all type of proteins. What are their types? What are the web servers? How you can access that? So all of those things would be covered. First of all, structural databases are quite very essential tools for all the crystallographic works. For example, you have isolated a particular protein molecule and you want to infer the structure. In that case, in the wet lab experiments, you have to go for XRD, you have to go for NMR to elucidate the structure. However, these structural bases are basically, an accessory technique or accessory thing that is available if you want to go for the structural prediction. However, you can basically this is a time-saving tool because the process of producing, solving, refining, and publishing the structure of new material is completely done within a short span of time. So the common information that is normally found in the structural databases, that is bibliographic information. Bibliographic information means which scientist or which author have worked over it, author name, the journal reference, what is the chemical nomenclature, the complete nomenclature detail, the formula, oxidation states, and all the elements present. Number of formula unit per cells is given, dimension and symmetry, whether this is 3D structure or in which dimension, what kind of cells would be present, what are the atomic coordinates. So all of these things will be covered in this common thing. So the basic structural database application is the structure refinement, classification, comparison, analysis, prediction, and database annotation. So we will be discussing it. These are the three main types, that is PDB, protein data bank, CATH, that is class, architecture, topology, and homology. SCOP means structural classification of protein. First of all, we will be discussing about PDB, that is protein data bank. This data bank, it basically contains the information about experimentally determined structure of proteins, nucleic acid, and complex assembly. That means, suppose you are working in a laboratory and you are working on a particular kind of protein. In that case, you go for entering the structure. After entering the structure, you will be basically depositing all the structural information depending upon your experiment into this database, so that any other scientist who is working in another part of the country or in another country of the world, in that case, he or she can be able to access that structural information for their reference work. So that is why this data bank is basically designed, in which the complete X-ray crystallographic data, NMR based data is deposited there. The main repositories are RCSB, PDB, PDBE, PDBJ, BMRP. These are the main repositories of protein structure data, in which the file format is basically PDB format, MMM-CIF format, PDB-ML, and XML format. So the first, let us discuss about more about PDB. That means all the 3D structure of all kind of protein is available in this database. The structural information is determined by what? XRD and NMR. However, this PDB complete this database, it is being maintained by Worldwide Protein Data Bank. That is why we call it as WWPDB. These are the few links of these sites, that this is the worldwide PDB link. This is the PDBE. PDBE means Europe site link. PDBJ means Japan site link. So each entry in this PDB is, again provided with a very unique identification number, accession number, just like that in case of GenBank also. In GenBank also there is a accession number, a unique number, so that you can go on identifying a particular kind of protein depending upon that. So this is the site of PDBE, that is Protein Data Bank Europe, in which you can see all text you can search. You can search in the PDB database or you can go for advanced search. However, I have pasted its link already in the previous slide, so you people can check on it. This is the complete entry point. That is the in case of advanced search you want to go. You can either mention the identification number, you can mention some text, You can add some detail, author name, journal name, if you know about it. All of these things can be mixed. Then experiment time. What kind of data you want? XRD, NMR, EM, or whether it is a protein molecule, DNA or RNA or some saccharide. So depending upon that, you can go for search. That means this advanced search is basically providing the comprehensive information about a particular protein, sequence info, domain info, sequence similarity, literature, and all the details apart from the structure is listed in this database. However, this database is also cross-referenced to SCOP and CATH, that are other your structural databases. This is the format that how these PDB based databases are basically designed. As we have discussed in our previous lectures also, that there the databases should be such that all the data that is contained in that very database, it should be in a particular standard format, that so that their uniformity can be maintained in a particular type of database. For example, in case of GenBank, the flat file format was there. So similarly, in the case of PDB, the PDB file format is there, which is the standard format for any protein structural file, so that it can give a detailed information about how the molecules are held together in 3D structure of a protein. It contains basically thousands and hundreds of lines, which we call it as records. As you can see, the file contains hundreds and thousands of lines called records, and each of the record will provide us a detailed information like header. Header will be providing us the information, file name, the date of submission, and PDB ID. So you can say what is the name of that protein for which you are looking for. That name would be listed. Date of submission, on which date the scientists have submitted that very entry, and the unique PDB identification number would be listed in the header. Then is title. It will contain the title of PDB entry, that is the complete name. Compound, this will include the protein name, that what is the name of the protein for which structure has been added. Then the source, this record or this protein has been isolated from which kind of organism or what is the source of it. So you have to mention the source. Then this is keywords. Keywords means it will contain the keywords that can give the best description of a particular protein. After that, EXPDTA, that means experimental data. It will give the detailed information that what kind of methods or experiments have been used for the elucidation of this protein structure. So the complete experimental data methodology would be mentioned. For example, you have carried out XRD. So you would be writing XRD or X-ray crystallography. From there, any person who is going to check on it, he or she would be able to understand that this structure is based upon this very method. Yeah, or this method was carried out for predicting the structure. Next is author name. That means it will list the name of contributors or name of the scientists who have worked in this area, who have generated this data so that it can be entered inside the database. Then is REV data. REV data means revision date. That means suppose in the first stretch you have made an entry inside the PDB and you have entered the detailed information. Suppose now you have repeated the experiment. At that time, what you got that there are some discrepancies or there are some mistakes, and you want to update your data. In that case, this is the revision data. That means all the revision date, that on which date you revised that data or updated the data, that complete thing would be mentioned. Next is JRNL, that is journal. That in which journal, in which journal details or volumes you can get the detailed literature about this protein. Remark, this will contain about brief remark or short remarks about the protein structure. And then is main DBREF, that means reference to the protein in the sequence databases. That means the same protein for which other scientists have worked and their sequences are basically submitted in different kind of databases. So this would be containing the reference. Next is SEQRES. It will contain the information about the amino acid sequence, that what is the sequence of that very protein, amino acid sequence. HET that will comprise of other non-proteinaceous molecules that are present along with that very protein. Then is HETNAM. It will contain the compound name of known protein substances. Then HETSYN, it will contain the information about other identical or analogous or homologous substances of non-protein molecules that are present along with these protein of interest. FORMUL, it will contain the chemical formula of the non-protein substances. Helix, it will contain the recognition of helical substructure, that where the helix is present. Okay. Next is link. It will, basically predict the structure of recognition of inter-residue bonds, that where are, from how these residues are internally bonded. Atom, it will contains the information about the atomic coordinates of the structure. Heat N, it will contain the atomic coordinated record of non-proteinaceous substances. Connect will contain the information about the bonds involved. Master will be containing the detail about the number of remark, HET, helix, and connect records and sequence records. And last line would be the representing the end of the file. So this is the kind of file that you get it. This is PDBE entry 1 CBS. As you can see, crystal structure. This is the your main title. This is the header. This is the method, experiment, release date, author name, primary citation, keywords, organism, uniprot. Everything is mentioned in detail in different types. Next is CATH. CATH means class, architecture, topology, and homologous superfamily database of protein. That means in this very database, you will be get to know the class of protein family, what is the architecture, what is the topological features, what is there any homologous superfamily of proteins present or not. In that case, this all these details will be present here. However, this complete database, it was developed at the University College London by Janet Thornton and some associated colleagues. These are the links of these CATH database. By clicking on it, we will be redirected to the database site. This is the CATH DB info. It is a main protein classification tool. Basically, it consists of four levels, that is class, architecture, topology, and homologous superfamily. So what the class will infer. Now, as the name suggests, C, the name of this database is derived from the first letter of these four levels. C corresponds to class, A corresponds to architecture, T corresponds to topology, and H corresponds to homologous. So class, it will comprise about the structural conformation of the proteins and their contents, that whether they are at this very, from this residue to this residue, there would be alpha helix, or there would be beta plated sheets, or there would be alpha beta regions. So all of those information would be confined in this class level. Then is architecture. It will briefly describe, or you can say it will provide us the gross orientation of secondary structure. That how the alpha, beta, and this complete secondary structure is assigned. It will also give us the complete detailed information about the folding of polypeptide chains also, that how many polypeptide chains are there and how are they being folded in a particular pattern so as to form a secondary structure. Next is topology. However, it deals with the structures formed due to different topological arrangements of secondary structure. It will explain, it will give you the brief description about the superfamily of the protein. Last is homologous superfamily. It will compare the sequence and structure of different proteins. That means you can go for integrated sequence and structure similarity search. Basically, this thing is mainly done if you want to trace the evolutionary relationship among the proteins. In that case, we go for homologous superfamily. So this is the one. As you can see, class, there are three major classes. Either the protein structure can be alpha helix, or it can be beta pleats, or it can be alpha beta. That means it is comprising both type of secondary structure. This is a class. After that, suppose we are taking this class, alpha beta. In that case, architecture. There are 32 architecture. Whether the proteins would be, folded in a pattern such like that they are forming some barrel-shaped structure, they are forming some sandwich-like structure, or whether it is three-layered or two-layered, as you can see. This is a barrel. This is a three-layer sandwich. One, two, three. There are three layers. Here you can see only two layers. After that, suppose you have taken this three-layer sandwich structure, and then we go for the topology. Then there can be different folding patterns. There are, many fold families from which you can infer that how these proteins are being folded to show to form a particular type of topology. Just a minute. Next is homologous superfamily. For example, lactate dehydrogenase. It is basically showing 4,000 different families depending upon the sequence. Then is phosphofructokinase. Then again, it is further divided into different types. So starting from the protein structure, you can go on inferring the complete homologous superfamily to which a particular protein of interest can belong. So the basic aim of CATH is to provide the official releases of protein structure within every 12 months. That means after every 12 months, the CATH database it gets updated. However, it is a freely available online resource. All time you can freely access it, and the main latest version it is fine to have reported or fine to have contained the information around 1,14,215 different domains, 2,178 homologous superfamilies, and 1,110 plus fold groups. That how are they being folded? This is the complete, when you click on the CAT database, this is the main home page that on how this, this is the brief introduction. These are the further links. These are the released versions, new data available. Now, this CAT have recently also set up a server that allows the users to submit the coordinates of the newly submitted structure so that automatic classification of CAT can be carried out. So the domain boundaries and sequence comparison boundaries can be, catered off. It contains also a very, significant detective program also, which is good for providing multi-domain proteins. Suppose there is a protein who is having many domains. So in that case, you can go on identification of those multiple domains. However, these results from this detective and this detective program, it is very fast. That means you have just entered the detail and you are getting the result within a time span of 1 minute. So all of these identified domains are scanned against non-identical representatives using a global sequence alignment method. That means you go for the similarity search also. If you are going for sequence similarity search, in that case, if the match is 95%, in that case we can say yes, this domain is identical to the protein that is available in the cat. However, if that similarity is less than 30%, then we say these structures can't be compared with all the sequence families. Then we have to go on searching for single sequence families for us to infer it. Then is how you can assess the structural similarity. There is a tool known as top scan. This top scan basically it compares the secondary structure in each fold family so that you can go on identification to which possible fold family my protein of interest would is showing some kind of similarity. So in that case you have to go for top scan. So each super family can be scanned off. Then after that there is a fastest version of this top scan that says SSAP that scans the representatives from all the times, all the families, and these structural pairs by using this SSAP score. If it is more than 80, then we say that these proteins are found to be homologous. If the scores are less than 70 to 80, that means we say there is no sequence or functional similarity. Finally, the complete SSAP-based structural alignment is displayed onto using a graphical display package that we can further, access. This is the complete distribution of SSAP score that I am discussing about. As you can see, these are the scores. This is the frequency. These are the different scores, and these are the results of SSAP run against. Suppose you have gone for the searching of a single kind of protein. In that case, all the superfamilies would be scanned, and these are the CATH code, and these are the SSAP score. As you can see, this is showing 90.68, 92.84. So if it is more than 80, then we can say yes, it is homologous, or yes, it is belonging to this very superfamily. If it is less than, then we can say no, it is not showing any kind of sequence similarity or structural similarity. So this kind of structural alignment main page is being displayed at the graphical display package. As you can see, this is the sequence, and this arrow it is basically showing the direction, and these colors these residues are marked by different colors: red color, blue color, green color. So all of these colors are basically showing the residue type, and such kind of structure, such kind of alignment is being shown so that the further alignment can be carried out. This was all about CATH. Now, next database, that is SCOP. The full form of SCOP is structural classification of proteins. Again, the structural classification of protein is abbreviated as SCOP. The first letter of each word is taken off and the short abbreviated form is being devised. Now, the structural classification of protein databases, it is basically done when you want to go for inferring sequence and structure of a particular database. This is the website link, as you can see. By clicking on it, you will be redirected to the SCOPE database homepage. Basically, it is a manual classification of protein structural domains, which is dependent upon the similarity of their structure and amino acid sequence. That means we go for sequence structure alignment. This complete database, it was created in the year of 1994 and it is being maintained at the Laboratory of Molecular Biology in Cambridge, England. It was devised by Mergen and his colleagues in the year 1994. Basic concept is this database is basically providing a detailed and comprehensive description of relationship of many known proteins. The classification is basically dependent on many hierarchical levels. However, this database is freely available. It is not paid. It is freely available online tool that you can anytime access it. This is being maintained. However, there are many prototypes. There are many released new versions also. From there, you can go on working. So this is the complete classification part of it, that is family, identical structure and function, that all proteins that have residue identities of 30% or greater wise or pairwise sequence similarity greater than 25%, those proteins would be categorized in a single family. If they are having lower sequence identity, whose structure and functions are very similar. So from there you can infer the clear evolutionary relationship. Next is superfamily, that is common structure and function. That means if a particular protein of interest have been found to be, arising from a common ancestor, in that case, the families whose proteins are having low sequence identity, they are having low sequence identity, but their structures are similar. Because their structures are similar to their functional features would be same. That is basically due to the common evolutionary ancestor because of the presence of, you can say, we have you must have read about this thing in immunoglobulin concept also. In that case also, there are different regions, variable and constant regions, due to which the different structures are being formed but their function remains the same. Fold is the main common core structure. That is the major inference of common major structural similarity, in which we go for inferring. We basically take the value of superfamilies and families, and they are defined to have a common fold if their proteins have same major secondary structure in the same arrangement with same topology connections. Next is class. That is, if there is similar secondary structure contents, then the folds have been divided into 5 structural classes. Either it can be all alpha, that means the particular protein structure, it is mainly comprised of alpha helix. Either it can be all beta, that means the structure is mainly composed of beta sheets. Or it can be alpha beta, that means alpha helix is also present, beta sheet is also present. Or it can be alpha plus beta, that means in which both alpha helix and beta strands, they are segregated, they are combined. Or it can be multi-domain, that in which different codes for which no homologs are reported till now, they are also being formed. So this is the structural part, alpha, alpha and beta, beta, which we have discussed in a previous slide also. Fold is tin barrel, sandwich or roll. And example is, as you can see in the case of sandwich, flavotoxin or beta lactamase, depending upon the folding pattern. This is the example of different type, monomer A and monomer B. This is the structure of interleukin 8. You can say, as we know, beta pleated sheets, there are two types. Either it can be parallel or it can be anti-parallel. So if they are running, the structure is being run in the same direction but in the alternating fashion, that is known as alternating parallel sheets. If they are, having some anti-parallel beta sheets but they are not alternating, in that case we call it as anti-parallel beta sheets. Now these are the main folds. Mainly alpha, they will be folding in bundle shaped or non-bundle shaped. If they are mainly composed of beta sheets, then it can be single, it can be roll, it can be barrel, it can be clam, sandwich, prism, solenoid. And if it is alpha or beta and alpha plus beta, there can be two possibilities. Either the structure can be closed or it can be open. If it is closed, then it can be barrel shaped or it can be roll shaped. If it is open, it can be sandwich shaped or clamp shaped. This is the main scope classification stats, that how many proteins are present, how many folds are present, how many superfamilies or families are present. So in this case, if all alpha proteins, if that means those proteins who are basically composed of alpha structure only, the number of folds are found to be 284, the number of superfamilies 507, and number of families is 870. In case of all beta proteins, it is 174, 354, 742, alpha and beta proteins, alpha plus beta proteins. There are multi-domain proteins. There are many transmembrane and cell surface proteins, small proteins, and in all, in nutshell, what are the total number of proteins. However, all of these proteins, the database, the SCOPE database, it is containing immense information about different kind of proteins and their structural information. From there, any scientist who is working any part of the world can access that information for their reference. This is the organization method, that how you can go on assessing. Enter the scope at the top of hierarchy and you can go for the keyword search. You can either use a PDB identifier or you can use some MRC site. However, if you have pre-searched it, you can retrieve that thing from the history also. It contains the information of all types of files. You can also enter if you want to go for the analysis. This is the main top. This is root. Scope is written. All beta, alpha proteins, all beta proteins, alpha and beta, multi-domain, membrane and cell surface, small protein, coil, and everything is written. Now you have to click on one. Suppose you have to, you want to search this site by using the search engine. Then you can enter the detail here and search the scope database for retrieving the information. So in that case, we have clicked on alpha and beta proteins. That means those proteins who have alpha and beta protein structures. So here you can say the what is the lineage, root was scope, class is alpha and beta proteins, and in which how many folds are present. Either it can be 10 beta, NADP binding, FADP binding, nucleotide binding, MURCD. So you have to click on any one. So in this case, I have clicked on 10 beta alpha value. In that case, the lineage would be root class fold. Now the fold part is done. Next is superfamily. What is a superfamily? That means in the case of 10 beta alpha viral, how many different proteins or how many superfamilies are present. In that case, you can see there are range of examples here. Triose phosphate isomerase, ribulose phosphate binding barrel, thiamine phosphate synthase, pyridoxine, FMN, inosine, PLP. You have to click on one. Suppose I have clicked on thiamine, or I have clicked on triose phosphate isomerase. In that case, the superfamilies are listed here. You can see triose phosphate isomerase in the case of chicken, in the case of human, rabbit, nematode, in the case of different organism, and these are the number of entries that we can check. This is in the case of ribulose phosphate binding value. You can see histidine biosynthesis enzyme. It is present in which case, in which kind of organism it is present. You can go and assess it. Suppose now you have selected one enzyme. That means you have selected one protein from a superfamily. Suppose that is, as you can see, this is the lineage. Family is histidine biosynthesis enzyme from ribulose phosphate binding barrel, in which protein is phosphoribosyl formino 5-aminodazole carboxamide riboyoid. Isomerase is A from Thermotoga maritima. I have taken the species this. From there, you can get the PDB entry domains, that where the structural information of this very protein isolated from this very organism is present. So here is the link of PDB entry, chain A, chain B. That means of two polypeptides, both of the links are given. So you can directly go on that and you can check out the structure. Now there are many scope mirror sites that are being maintained at different sites all around the world. For example, in UK, the main home server is in Cambridge. This is the website. USA, University of California, Australia, Japan, Taiwan, Singapore. In India is also present. That is Madurai Kamraj University, or Indian Institute of Science, Bangalore, IISc, or CDFD, Hyderabad. So all of these are sites linked for which you can go on assessing all of these scope mirror sites in case of Russia, case of Israel, China, or Italy, and all of these mirror sites are mentioned. From there, you can go on check. You go on check all of these scope sites. Now, the main relevance of the SCOP databases in bioinformatics, we can use this classification as a reference set of data if you want to develop some automatic classification methods from which you can go on analyzing any family, any superfamily, and food. You can also build an integrative structural data mining tool so as to predict the methods and structure comparison tool. You can go for understanding the evolution of protein enzymatic functions, their evolutionary change, their structural evolution, how you can correlate the distantly related proteins depending upon their same fold. You can go on studying structure and sequence variability and its dependency on homologous proteins. You can even derive amino acid similarities matrices such as substitution tables, substitution matrices, for example, PAM, BLOSSOM. So all of these can be input. After that, it have been used to carry out many combinations of different domains and different decomposition and multi-domain proteins also. These have been used for developing many kind of value-added and more specialized databases also. So the scope is immense and you can use this thing in many ways. This is the further use. Experimental structural biologists can use this database so as to explore the region of structure space. Molecular biologists can use so as to locate the protein of interest and their links make exploration easy. Theoretical scientists can make use of it. Scope will be, it is a kind of pedagogical use for it organizing structure in a very comprehensive manner. You can go on searching for particular families, you can go on searching or you can ensure the growth of structural data so at that give you an estimate of total number of protein codes and number of superfamilies that are present in the nature. So it provides a level of classification that is not present in the pdb. So that was all for today. Thank you so much.